I was recruited back from Yale in July of 1968 after the terrible series of riots that occurred in Washington. We started anew in the basement of St. Elizabeth's Hospital. Mimo Costa was recruited back from Columbia, so the two of us were each independent lab chiefs working side by side on 2 floors of the place, and it was a big step up in what we were able to do. We were able to build a pretty good laboratory system there. Several of the postdocs who had been selected by Sel Maraghi stayed on with me and have become very famous people in their own right. Roger Nickel, Barry Hoffer, George Siggins. We were able to do a very extensive analysis of how does norepinephrine work, and because we were working next to Costa's lab, where they were working on second messenger transduction, we were able to take their biochemical findings and immediately put them to the test with the electrophysiology. We were able to develop the Swedish method for fluorescence histochemistry and combine the electron microscopy with the light microscopy. We started, I think, immunocytochemistry in a major way with antibodies to cyclic AMP, which I was able to get from one of my previous teachers at Washington University, Charlie Parker. It seemed like an ideal place to work. And then government started becoming somewhat less pleasant. A lot of regulations, a lot of travel stipulations, a lot of pressure not to do things for money while you were there. When I was at Yale, shortly before I came back to St. Elizabeth's, Nick German had been in a car accident, and as a result of those injuries died. He had signed a contract to do a book for Oxford University Press, which was the course that Nick and Jack Cooper and Bob Roth and I taught to the first-year graduate students. And so to dedicate his memory, we went ahead and finished the book. And they didn't want me to go back to his funeral. I had to get special permission to leave Washington to go back for Nick's funeral and to work on the book. And I couldn't work on the book in my own time. Every time there was a new edition, it was another series of hassles. We had no idea that writing a small, irreverent paperback would become a lasting monument to our dear friend. But I think it's a nice icon of how the field has grown and matured. 1975, I started being open-eared to invitations to take a look at other places. Mm-hmm. Normally when someone would say, what would it take to get you to come do your stuff at our place, I would say, well, it'd probably take $1 million. And they would say, oh, and go on their business, and I would go on mine. And one day, Frederick D. Hoffman, who was the president of the Salk Institute, asked me that question, and I gave him that answer. And he said, I'll call you back next week. He called me back the next week, and he had half of the money. He said he felt sure that if I went with him to a couple of places that we could get the rest of it. And he was true to his word. Jonas was in Washington, and I interviewed with Jonas and went out and looked at the place and gave my job seminar. And they agreed to move a major part of my lab from NIMH there to set us up with the equipment that we need and build us a really splendid laboratory. And so we moved at the end of 1975, and the labs were up and running in the new location by July of 76. In that interval, my next door neighbor at the Salk, Roger Guillemin, had discovered a couple of the endorphin peptides. In fact, while I was driving across the country, out came the paper by Hughes and Kosterlitz on the discovery of metenkephalin and leuenkephalin. I read those papers when I got there, and Roger said to me, we've just discovered another one of these peptides, but I don't know how to test them. And so one of the things I was really good at was making intracisternal injections. So we had a ball for that first 6 months without a lab, just working sort of in Roger's space and scrap space around the Institute, injecting these newly synthesized peptides and watching what happened to the animals. Mm-hmm. I've always been obsessed with that initial question, what does norepinephrine do? We had a couple of insurmountable problems at the beginning. We didn't know where the cell bodies were that made the fibers. And so it was very hard to envision the actions of norepinephrine in terms of circuitry. It was only possible to look in terms of postsynaptic pharmacology. And being able to show that a major part of its action could be mediated by cyclic AMP generation postsynaptically. In that era, it was just about the time when Earl Sutherland won the Nobel Prize for the discovery of cyclic AMP and second messengers. To be able to show that the nervous system exploited that same kind of thing was very exciting. But we spent a lot of time working with other postdocs, devising methods initially started by Ed Evarts to record from the brains of freely moving cats to identify the likely source of the norepinephrine fibers. And by the early 70s, we knew where that was. And to record from the locus coeruleus and the raphe during free spontaneous behavior. And that made it very clear that their role was a kind of regulatory role on cortical arousal. It followed the origin and ending of the sleep stages. And so putting those systems together, one began to get a kind of global view of how a Pontine system that projected to a lot of places could modify the action of those places in a way that might then lead to the enduring changes that you could learn from. And we ran the gamut with the norepinephrine system, studying its development, studying the origin of its synaptic inputs and outputs. And so I would say, without a question, it's sort of the characterization of the brain norepinephrine system and its actions that stands out in my mind as what we always wanted to do from the beginning, and only 10 years later, when the tools were there, were we able to put it together. And it became the sort of paradigm for what we wanted to do with the peptide systems once we could see them. So when the neuropeptides became the focus of attention, then the same questions arose. Where are the cell bodies? What happens to the postsynaptic cells when you stimulate the pathways that have the peptides in them? And that's how, when we started with the opioid peptides, that's exactly the paradigm that we tried to follow. In the late 70s, 1979, a paper came out in Nature which completely revolutionized the way people found neuropeptide systems. The classic way that Roger Guillemin had done it, starting with 750,000 sheep hypothalami and extracting it and doing bioassays, was a major tour de force, and that's a major basis of their Nobel Prize recognition. But molecular biology was finally becoming a laboratory art instead of something that people were afraid of. So cloning genes now was something that people were really investing in, and the technology was moving quite along. In March 1979, the group of Professor Numa in Kyoto published in Nature 10 pituitaries. They were able to extract enough messenger RNAs and clone them to identify the entire precursor of the beta endorphin ACTH common precursor. And buried in that molecule was what looked to be yet a third hormone with ACTH and beta endorphin. And within a week, Dr. Guillemin's lab had synthesized that, and we were able to spritz it on, make antibodies to it, and map it out. And it seemed like, well, now this technology would mean that if there are a lot more peptides, and that's what everybody thought, this would be the way to go at it. So I went over to the Scripps Clinic, where I knew Richard Lerner, and he introduced me to his postdoc, Greg Sutcliffe, and I said, how hard is what they did to do? And they always said it's not hard at all. I said, well, why couldn't we just take the messenger RNAs from the brain and see, and look for the ones that haven't been discovered yet? How hard would that be to do? And they said, oh, it's a lot of work, but it's not that hard. So we started a collaboration in mid-1979, and from that point on, I was really knee-deep into the use of molecular biology for discovery, for the acquisition of in situ hybridization, so you didn't have to wait for antibodies to map out where the cells were, and then to take those gene products and try to understand if they were really neuropeptides or they were some other kinds of signals. And it led us to whole classes of molecules that really had not been envisioned before, and that kind of open-ended, mentally unconstrained attitude for discovery is what to me is still very exciting about the use of molecular technology to probe the structure and function of the brain. If you could discover new neurotransmitters, then you had some additional indices to use on measuring spinal fluids or brain tissues from people who died with mental illness, and maybe now we would find the one that's really involved in one of these situations.